Good morning, students. Today we are going to discuss about the cloning vectors. Actually, why we are going to discuss about the cloning vectors? First, we should know. Actually, in the biotechnology, what we do, we first of all we identify a desired gene in any plant cell, bacterial cell, fungal cell, anyone, and then we isolate that desired gene from that bacteria or fungi or plant or animal. After that, what we do, we now link that alien piece of DNA or desired gene into a cloning vector for its cloning or for its multiplication to increase their number, because we requires the large number of its copy. Large number of the copy of that desired gene or that alien piece of DNA we require, because we are going to produce the large scale production of industrial product. So we require the large number of the copies of that desired gene or alien piece of DNA. So it requires the cloning vector. Cloning vector means the vectors in which we can link that alien piece of DNA for its replication or for its cloning, we can say, or to increase their number. We know that both plasmids and bacteriophages, actually these can be used as the cloning vectors. So that's why we are going to discuss about these plasmids and bacteriophages. And we know that both plasmids and bacteriophages replicate in the bacterial cell, without the use, without the control of the chromosomal DNA of the bacteria. Means they are independently replicate without the control of the chromosomal DNA of that bacteria. It means both can be used as the cloning vector. Both plasmids and bacteriophages can be used. We can use as a cloning vector. Now we will talk one by one. Suppose what is plasmid? Suppose this is the bacteria. Suppose this one is the bacteria. In the bacteria, we know that there is a chromosomal DNA. Suppose this is the chromosomal DNA. Beside this chromosomal DNA, there is also found the extra chromosomal DNA, and this extra chromosomal DNA is called the plasmid DNA. And this plasmid DNA replicate independent of this chromosomal DNA of that bacteria. So, when we link any alien piece of DNA with this plasmid DNA, we know that it has the capacity to replicate itself. It can independently replicate in the bacterial cell. So if we are able to link any alien piece of DNA with that plasmid DNA, it replicate in the bacterial cell, increase the number of the desired gene also. That is called the cloning of that desired gene or alien piece of DNA. Plasmids in some bacteria, actually in all bacteria there is no plasmids, but in certain bacteria there is the extra chromosomal DNA found, that is called the plasmid DNA, and it has ability to replicate independently of the chromosomal DNA. So we can add the alien piece of DNA or the desired piece of DNA with this plasmid DNA, and we can increase the number of the alien piece of DNA. Because when this plasmid replicate independently of this chromosomal DNA, the alien piece of DNA would be also replicated. So their number also increases. Question is, why to increase the number of this alien piece of DNA or desired gene? Because we are going to produce the large scale production, industrial production of any product. So we requires the large number of the desired genes or alien piece of DNA. So these plasmids can be used as a cloning vector. But the thing is, in some bacteria, the copy number of the plasmid are 1 or 2. In some bacteria, the copy number of the plasmid DNA is 1 or 2. While there are also certain bacteria, the copy number of plasmid is 15 to 100. 15 to 100 we can say. So there are also certain bacteria, the copy number of plasmid means the per cell, the number of plasmid are 15 to 100. There are also some bacteria. While in some bacteria, the copy number of the plasmid or number of plasmids per cell is 1 or 2. While in some bacteria, there may be more than in some bacteria, there is the 15 to 100 copy number per cell. It may be even larger also, more also. Similarly, now we will talk about the bacteriophages. We know that suppose this is a bacteria, this is a chromosomal DNA of bacteria, this is a chromosomal DNA. Suppose a bacteriophage, suppose this is a bacteriophage, when this is the DNA of bacteriophage, when a bacteriophage infect a bacterial cell, its DNA enter into the bacterial cell. And now after that, the DNA now incorporated with the chromosomal DNA also and replicate along with the chromosomal DNA also and use the machinery of the cell of that bacteria and produce the large number of phage viruses, per cell in the bacteria. Thousands of bacteriophages are produced during the multiplication or reproduction of the bacteriophage. So per cell, large number, thousands of bacteriophages are produced per cell. So if we are able to add a alien piece of DNA or desired piece of DNA with the DNA of this bacteriophage, we can increase their number equal to the number of bacteriophages that produced per cell in that bacteria. So there we know that per bacterial cell, the number of bacteriophage is very high. It is very high. So large number of when the bacteriophage infect a bacteria, its DNA enter into the bacterial cell or host cell and produce use the machinery of that cell, use the ribosome of that cell, synthesize its own protein and produce the large number of viruses. Thousands of viruses are produced per cell. So because the number of viruses of bacteriophage is very high per cell, so the number of its genome or DNA would be also very high. So if we add any alien piece of DNA or desired DNA with the DNA of that bacteriophage, it can multiply in large number per bacterial cell equal to the number of bacteriophages produced per cell. That is the copy number, we can say the copy number of the bacteriophages. So if one asks to you, suppose this is the question, also it is the question asked in the board also, that which one is the better cloning vector, plasmids or bacteriophage? We can say that the bacteriophage are the better cloning vector because its copy number is very high. Means if alien piece of DNA, if we are able to link with the DNA of the bacteriophage, we can produce, it can multiply in large number per bacterial cell. Means we can clone that alien piece of DNA in large number per bacterial cell. If we are able to link or add or join an alien piece of DNA with the bacteriophage DNA. So we can say that the bacteriophages are the better cloning vector because its per cell copy number is very high. While the plasmids, in many bacteria the number of plasmids are 1 or 2, while in some bacteria there may be the 15 to 100, it may be more also. But in the case of bacteriophage, the number is very high. When a bacteriophage infect a bacterial cell, it multiply and reproduce in that bacterial cell and produces large number of its copy. So that alien piece of DNA will also produce in large number. So we can say that out of these two cloning vectors, plasmids and bacteriophages, we can say that bacteriophages are the better cloning vector because it's very high copy number per cell, we can say. Thank you.